Welcome to CarlChem. I'm Lainey, and in today's journal club, I'm going to talk about this paper titled De Novo Design Proteins Neutralize Lethal Snake Venom Toxins. This is a collaborative paper from a couple schools in the US, Denmark, and the UK. As always, this is an open access article, and the DOI link is in the description. There are two major pieces of this article. Number one is how the authors used computational approaches to try and make a broad spectrum snake antivenom, and number two is how the antivenom protein candidates perform in vivo. The scientific need for this paper is that snakebite venom causes 100,000 fatalities and 300,000 permanent disabilities annually. In 2017, it was considered the highest priority neglected tropical disease by the World Health Organization. This paper focuses on a treatment for three-finger toxins, which are a part of elipid snake venom that's life-threatening. A little bit of context here. Elipids are a family of snakes that always have their fangs out and they're usually venomous, so they're pretty much always ready to throw down. This family includes king cobras and black mambas. Most Elapidae venom will have toxins from either the PLA2 or the three-finger toxin class. This paper is focusing on the latter. The only treatments for snake bites right now are using polyclonal antibodies derived from plasma of immunized animals. This isn't ideal because getting a treatment in this way has limited efficacy for three-finger toxins, it's expensive, and it's not a very efficient way to make a drug. The author's solution to this problem is to use computational methods to design a small protein that will bind to multiple toxins in the three-finger toxin family. They made and then tested this protein design in mice and found that their therapeutic candidates were successful in protecting the mice from a lethal dose of the three-finger toxin. To design the drug candidates, the authors used a deep learning-based RF diffusion method. This means that they're using computational models to create a protein structure by looking at selected properties like binding targets and designing the protein around that. Alpha neurotoxins are a prominent subclass of three-finger toxins. They consist of a small hydrophobic core held together by highly conserved disulfide bridges and three beta sheet loops forming the three fingers. You can divide the alpha neurotoxin family into short-chain and long-chain alpha neurotoxins, which differ by their length and number of disulfide bonds. They both bind to types of nicotinic acetylcholine receptors, or NACHRs, but despite their structural similarities, they have different pharmacological effects. The goal here is to be able to neutralize both types of alpha neurotoxins for a solid therapeutic effect. The current therapeutics focus on binding to the alpha neurotoxin in the NACHR binding site, but instead of targeting that binding site, the authors are targeting the edge beta strands of the fingers by making geometrically matching beta strands in their protein and relying on the size of their binder to physically block the venom from binding to its target. They did this with RF diffusion, looking at secondary structure, and took each of the three fingers into consideration individually. So the first thing they're looking for is to get a complementary shape and charge to fit into the alpha neurotoxin. Then they made the protein sequence that would fit that desired structure using protein MPNN, which is another deep learning algorithm, and they put that sequence into AlphaFold 2 to get the 3D structure of the protein candidates. After some filtering, the authors selected their top candidates based on the protein's ability to interfere with toxin binding to NACHRs. This was guided by more computational work that looks at the neurotoxin acetylcholine receptor complex with and without the binder they selected. Now that we have the design, we can get into the wet lab experimentation. Here, the authors assessed different drug designs and screened them using a yeast surface display and representative short-chain alpha neurotoxin. What this means is that they modified yeast cells to express the drug on their cell surface, then introduced the representative short-chain alpha neurotoxin and looked to see which candidate bound to it best. From this experiment, they selected the best protein by looking at binding affinity, or KD. In this case, the authors want a very low KD, which indicates binding at low concentration of their drug. In practice, this means stronger binding affinity. Their best candidate was SHRT, which I'm going to read here as short, with a KD measured at 0.9 nanomolar in SPR and 0.7 nanomolar by BLI. The properties of their short protein show a single peak in the SEC, typical CD indicating normal alpha helices and beta sheets, and a melting temp of 78 C. Looking at the data here, the SEC is going to separate based on size. For this data, the SEC is telling us the purity of their drug is 100% because there's only one peak. This peak is super broad and has some tailing, which could be due to surface charge, or it could be encompassing multiple species. For early stage characterization, this is fine, but once you get farther along with this drug, you're going to have to be more convincing about the purity. A nice, easy way to complement this would be a gel or capillary electrophoresis. SPR is used to look at binding kinetics. Here they bound the neurotoxin to the sensor chip, then introduce the drug, and then rinse. So what you see is the binding, then the dissociation. They found here that the KD of short is 0.9 nanomolars. For the BLI, which is very similar to SPR, we're seeing an overlay of multiple concentrations and a really similar result of 0.7 nanomolar for their KD. Really great to see a complementary technique here that confirms the results, especially because there's usually pretty high variability with this type of test method. CD here is used to look at the secondary structure. The negative peaks around 208 and 222 indicate alpha helices, and the negative peak around 218 indicates beta sheets, meaning the binder that the authors made has both alpha helical and beta sheet properties in its structure, which matches the expectation from computational data. Melting temp was also measured and shown to be 78C, which is good. It's thermally stable and shouldn't have any issues at body temp. The authors also did X-ray crystallography to look at the structure in the apo state, so in the absence of a ligand. Again, the results match the expectation from computational design. And from this X-ray crystallography data, we can see that short is binding to loop 3 of the neurotoxin, which is key for binding to muscle type NACHRs. What's happening on a molecular level is that the beta sheet portion of short is forming extensive hydrogen bonding with a complementary edge beta strand in the toxin. And tyrosine 45 of short forms a backbone hydrogen bond to cysteine 41 on the neurotoxin. Keep in mind this is still theoretical because the ligand wasn't bound during this experiment. To cover the class of long-chain alpha neurotoxins, the authors used alpha cobratoxin, which is very well characterized. They did the same approach as with the short-chain alpha neurotoxins, where they looked at binding affinity to choose the best candidate and found that their best protein was LNG, which I'm going to read as long. For the SEC, looks like they have a little more tailing on this guy, but I have the same comments as with the other SEC we saw. It's showing 100% purity for now. The SPR is showing a KD of 1.9 nanomolar, while the BLI says 6.7 nanomolar. When you're looking at these results, consider the order of magnitude that we're on. Since it's nanomolar, the difference between 1.9 and 6.7 isn't all that much for this type of testing, so the results do agree. And they aren't all that different from the short protein either. The CD is showing a similar trace to the short protein, but with an even higher TM of 95C. The authors also did X-ray crystal structure here, but for long they characterized the protein with the alpha cobratoxin bound. The measurements that they got agreed with the computational data pretty well, and similarly to the short, the long protein is using a complementary beta sheet portion of itself to bind to a beta sheet portion of the toxin's fingers. In this case, it's binding to loop 2, which is crucial for binding to muscle type and neuronal alpha 7 NACHRs. They're also seeing hydrogen bonding between the backbone of long and the alpha cobratoxin arginine 33, which is at the top of loop 2. Next up, we're looking at the design of cytotoxin binding proteins. Cytotoxins are another prominent functional group in the three-finger toxin family, and as their name suggests, they have cytotoxic effects. They do this by destabilizing lipid membranes. These types of toxins need to be addressed because if left untreated, cytotoxins will cause patients to have limb deformity, require amputation, or have lasting disability from the snake bite. When designing a binder for cytotoxins, the authors wanted to steer away from the beta strand binding because it may not be enough to prevent the membrane disruption caused by these cytotoxins. Instead, they focus on binding directly to the three-finger loops of the cytotoxin by creating hotspots in their protein that will bind to the tips of the fingers of the cytotoxin. The authors did a similar screening as the other two proteins, but this time they also tested a luminescent cell viability assay. In these results, the top candidate was CYTX, which I'm going to read as Cytox. Neutralization results are in supplemental figure 5, where we see cell viability for controls with no cytox and with a ratio of 1 toxin to 5 binder. We can see low viability for untreated controls and good viability at about 70 to 80% in treated samples. They did the same characterization testing on Cytox as well. SEC looks similar to the short and long proteins, and SPR is showing a KD of 271 nanomolar for Nia-Pellida whole venom. The SPR looks really different from the other two proteins. You can really see how much larger the KD is here because of those drops you see during the washing step. The CD agrees with the other two proteins and the computational assessment. We have both alpha helical and beta sheet secondary structure here, and the TM is the lowest at 61C, but still totally suitable for human body temp of 37C. The authors also did a crystal structure for Cytox, and this was bound to Nia-Pellida cytotoxin. To get better crystallization, they had to add in a disulfide bond to connect the beta sheet segments of the two alpha helices. It looks nearly the same as the original, but with a slightly higher TM of 70.3 and a higher KD of 740 nanomolar, meaning it's less efficient binding to the Nia-Pellida cytotoxin than without the disulfide bond. The crystal structure of the Cytox B10, which has the extra disulfide bond, shows that we're binding using the hydrogen bonding in loops 3 and 2 of the cytotoxin on the left insert, and on the right side showing a salt bridge between asparagine 57 of the Cytox and lysine 18 of the cytotoxin. These results match predictions from the computational model as expected. The authors moved on to in vitro neutralization experiments. For the alpha neurotoxins, the authors used patch clamp experiments to assess drug efficacy using a human-derived cell line that expresses muscle-type NACHRs. In a patch clamp experiment, you take a very small pipette and attach it to a cell membrane by light suction, which forms a tight seal and allows you to look at a very small portion of the membrane. The pipette contains some electrolyte solution so you can apply voltage to it, and then you measure the resulting current. Looking at the result for the current, you can tell if your transmembrane ion channel is open or closed. In the context of this experiment, we know that the NACHRs will open when bound to acetylcholine, and we know that the alpha neurotoxins will bind antagonistically to the NACHRs, meaning that they bind to the receptor without producing a response and also prevent the target agonist, acetylcholine, from binding, which would keep them closed. If the venom is bound to the receptor, we won't see a current through the ion channel, but if the drug is working and acetylcholine is bound, then we will see a current. For the patch clamp experiments in this paper, the authors measured the current when acetylcholine was added to the sample to determine the maximum response. Then they added a solution of pre-incubated alpha neurotoxin at the IC80 concentration, meaning it's expected to inhibit 80% of the target receptor. This pre-incubated solution will contain acetylcholine, venom, and either the corresponding binder or an alternative nanobody treatment, which was published in 2023. If the short and long binders are successful, then they'll bind to the venom and allow the acetylcholine to bind to its receptor. Experimentally, if this works, we expect to see a current that's consistent with the acetylcholine control. If unsuccessful, then the current will decrease because the alpha neurotoxin is binding to the target receptor. In figure 4a, we have the short-chain alpha neurotoxin SC and TX experiment with its corresponding binder short. We can see that the short binder is consistent with the acetylcholine control at a ratio as low as 1 short to 1 toxin, meaning that a 1-to-1 ratio appears to be enough to neutralize the toxin in this particular experiment. The nanobody that we're using as a comparator doesn't show neutralization until a ratio of 1-to-27. In 4b, we see the long-chain alpha neurotoxin, alpha-cobratoxin, and its corresponding binder long. We're seeing a similar pattern as with the short binder. The authors say a 1-to-1 ratio is neutralizing here. I would err a little more conservatively and say 1-to-3, but it's still pretty good. Results for the nanobody are inferior to the protein in this case as well. In both charts, we see the negative control of a nanobody that has a different target, and we see that this has no effect on toxin neutralization. These results look promising, but they are in vitro, and their proteins had some time to bind to the neurotoxin before being introduced to the cells. I'm curious to know if this would still work if they added the binder after neurotoxin administration, because realistically, nobody is taking this anti-venom until after they're bitten. The authors used a cytotoxicity assay to assess their cytotoxin binder, Cytox. This is a different experimental design to the alpha neurotoxins because cytotoxins have a different mechanism. If something's cytotoxic, it will kill a cell, so the measurement here is cell viability. The authors pre-incubated 7 different Naya snake venoms with Cytox binder at 5 times the venom concentration, and then introduced this solution to cells and assessed viability. Since the authors had to use whole venom and not a purified toxin, they had to plan their experiment so that the results could be compared. Results here are normalized by using 2 times the IC50 concentration for each venom. The IC50 value corresponds to how much of the venom you need to get 50% of the maximum effect. The authors used 2 times this amount to try and see 100% cytotoxicity without the binder. They also estimated that the venom consists of about 70% cytotoxins, which is based off of proteomic analysis. They had to do this since they're using whole venom here and not a purified version. In the results, we can see the positive control using just cell media has 100% viability. The negative control using Triton has no viability. Triton is a detergent that's going to break apart cell membranes, so this is by design. Just the binder without snake venom also shows good viability. Not quite 100%, but cell viability is going to have variability, so I'm not surprised. These controls tell us that the experiment is designed correctly. We were able to tell when the cells were no longer viable, and that the binder does not appear to have any negative effects on the cells. Then they show each of the Naya venoms that were assessed without and with the binder. Without the binder, we see no viability, and with the binder, we're seeing about 70 to 90% viability. Quite a stark difference, which is implying that the Cytox binder is working. Unfortunately, even at the 1:5 ratio of venom to Cytox, intradermal venom administration in mice still showed dermonecrotic lesions, meaning that the Cytox design needs some work before it can get further testing. Since the alpha neurotoxin treatment showed good promise, the authors moved to an in vivo study using mice. They have a nice experimental design with a few variables. Mice were treated with lethal doses of neurotoxin, and their survival for 24 hours was monitored. In the cohorts that received treatment, a toxin to binder ratio of either 1:10 or 1:5 was administered. First, we have control groups treated with just the short-chain neurotoxin or alpha-cobra toxin. Results show 0% survival, which confirms the effects of these toxins. Then we have the cross treatment, so that's a treatment mismatch where the short-chain neurotoxin is treated with the long-chain treatment, LNG. Additionally, the long-chain neurotoxin, alpha-cobra toxin, is treated with the short-chain treatment, short. Unsurprisingly, these mice also had a 0% survival, which shows the specificity of the drug candidates. The last control we have is injecting just the short and long binders into the mice. No toxins here. This resulted in no negative effects for up to 2 weeks post-injection. Now we move on to the treatment variables. First is the pre-incubated treatment, where the short-chain neurotoxin was treated with the correct binder, short, and the alpha-cobra toxin was also treated with the correct binder, long. The binder plus toxin was incubated for 30 minutes, then injected into the mouse. Here we're seeing a 100% survival rate. This is really promising data, and it allows the authors to move on to the next step of their experimentation, which is a more real-world scenario. Now we have a more representative treatment where the mouse gets injected with either the short or long-chain neurotoxin and then receives binder treatment at a ratio of 1:10, 15 minutes after administration. Here we're still seeing a 100% survival rate in these mice. Then they waited 30 minutes to treat the mice. For the short-chain neurotoxin, we see 100% survival, and for the long-chain neurotoxin, we see 60% survival. The authors also note here that all of the surviving mice show no evidence of limb or respiratory paralysis. Lastly, the authors performed the same experiment, but using a toxin to binder ratio of 1:5 and administered it 15 minutes after toxin exposure. Here we see 100% survival on the short binder and about 80% survival on the long binder. These results are certainly promising, but I'd really like to see some context here on how the current antibody treatment is performing and how long you have to administer it. I'd really like to see some context for this experiment so that we know how they stack up to each other. In summary, the authors have created anti-venom candidates by using a computational approach to drug design instead of the current standard of immunizing animals. This is a more cost-effective and reproducible way to make anti-venom, but is still in early stages, so it won't be treating humans anytime soon. This research is really useful for the field overall because it's showing how powerful a computational-based approach to drug discovery can be. And the authors note that this in silico approach is less resource-intensive than traditional antibody development and could help enable researchers in low-income and middle-income countries to better contribute to the development of effective treatments for snake bites and other neglected tropical diseases. All right, time for my comments. I particularly liked that they were binding to a conserved region of the toxins and relying on the physical bulkiness of the binder to prevent those toxins from binding to their intended targets. Really nice alternative to the specificity of an antibody, which isn't going to give you that broad spectrum efficacy. It seems like these protein binders for various alpha neurotoxins are promising, but they have limited testing in animals, which is completely expected at this stage of the research. It's a good starting approach to work on the representative toxins, but we would really need more data with other types of alpha neurotoxins to be able to consider them a broad spectrum approach, which is what the authors are trying to sell. I'm surprised they didn't include an in silico immunogenicity assessment since they already did a lot of computational work, but maybe that one gets included in a later paper. I'm very curious to see how they plan on formulating this because if you need administration via injection within 15 minutes of being bitten, it presents unique formulation challenges. They would have to have a portable device that's stable in the heat of these snakes' habitats, and it can be really difficult to prevent protein aggregation in heat and with shaking, even if you have a high TM. It would also need to be cheap enough to mass produce so that people could carry it around as a precautionary measure. I imagine in an ideal situation, it would have to be like an EpiPen, but for snake bites. All in all, I'm excited to see what they come out with next. I thought it was a really cool paper. What do you think? Are you convinced by these results? Let me know in the comments. And of course, like and subscribe for more.